Usually when we talk about a medical diagnosis, there's this expectation of precision for you as a patient,
Yeah,
right?
you expect a clear answer.
Exactly. Like, you break your arm, the x-ray shows a jagged white line, and the orthopedic surgeon points right to the fracture. It's localized, it's, clear cut.
But biology rarely works like that at the cellular level.
It really doesn't. And today, we are stepping into a biological mystery that totally defies that kind of clean structural logic. I want you to imagine a vital manufacturing center, let's say, the facility that produces all the specialized components, keeping a massive city functioning.
Okay, setting the scene.
Right. So suddenly, a specific assembly line goes completely rogue. The units start overproducing at this exponential rate. But worse, these units are defective.
Yeah, they aren't just useless, they're actively harmful.
Exactly. They start leaking these toxic byproducts that actively dissolve the factory's infrastructure. They start replacing the delicate machinery with, thick, dense concrete.
It's a complete structural collapse.
Yes. Eventually, the facility is completely choked off, buried in its own concrete, yet somehow it's still frantically churning out defective parts.
It's a cascading system failure. And honestly, it perfectly mirrors the pathology we are unpacking today for you. We are looking directly at the human bone marrow.
Well, the primary site of hematopoiesis, right, where all the cellular components of your blood are continuously generated.
Exactly. And to figure out how to stop this cascading failure, our mission today is to pull from a really fascinating stack of research.
Yeah, primarily a groundbreaking publication from the journal Cancer Cell. We're doing a deep dive into a hidden epigenetic network that dictates lineage commitment in blood cells.
And more importantly, we're looking at how researchers identified a specific master regulatory protein that acts as this, profound Achilles heel for severe blood disorders.
The source material is incredibly dense but clinically vital. We're talking advanced literature on megakaryopoiesis, examining the pathogenesis of myeloproliferative neoplasms, or MPNs, and looking at highly specific genetic leukemias.
We also have clinical trial data to go through, specifically from the Augment 101 studies, which evaluates a novel class of targeted drugs called N inhibitors.
But before we can isolate the disease, that rogue factory pouring concrete over its own floor, we need to establish the baseline for you. What does normal look like?
Right, the normal operating procedure of the marrow. We should start with this central player in this biological drama.
The megakaryocyte.
Yeah.
And the process of megakaryopoiesis. Let's break that down word by word.
Good idea. So megakaryopoiesis is the developmental pathway that produces megakaryocytes. Mega meaning large, karyo referring to the nucleus, and poiesis meaning to make.
So making cells with giant nuclei.
Exactly. These are the largest cells in the bone marrow, and their developmental life cycle is entirely unique. Most somatic cells progress through a standard cycle. They replicate their DNA and then undergo cytokinesis, physically dividing into two distinct cells.
But megakaryocytes don't do that, do they?
No, they bypass cytokinesis entirely. They undergo a process called endomitosis.
Okay, wait. Let me use an analogy here for the listener. Think of a megakaryocyte as a giant water balloon. So with endomitosis, they are replicating their entire genome, duplicating the blueprints, but they never actually split the balloon into two.
Precisely. They just keep accumulating genetic material inside the same balloon. This is a process of polyploidization, or ploidy.
Right.
While a normal cell is 2N, containing two sets of chromosomes, a mature megakaryocyte can reach 64N, or even 128N.
Oh, wow. That's massive.
It becomes a massive multilobulated warehouse of genetic blueprints and raw materials. And when it finally reaches full maturity, it extends these long branching structures called proplatelets directly into the marrow's blood vessels.
And then the sheer physical force of the flowing blood just rips those extensions apart, right? It fragments them into thousands of tiny pieces.
Exactly. Those tiny fragmented pieces are platelets, the crucial components that clot your blood.
So let me push back on this, or ask a question you might be wondering. Ploidy means the cell is just hoarding copies of its own blueprints without actually splitting. Why would a cell do that instead of just reproducing normally?
It's a brilliant evolutionary adaptation when you think about the biological cost of production.
Because if you need to rapidly deploy millions of platelets to stop bleeding after a severe trauma, relying on standard one-to-one cellular division is just, it's far too slow.
Right. The megakaryocyte acts like a massive localized server farm. By caching an enormous amount of mRNA and protein machinery in one giant cell, the marrow can instantly deploy thousands of clotting packets on demand.
Rather than waiting for individual cells to replicate from scratch, it is the ultimate biological optimization.
It is. It's designed for massive rapid output, but that exact optimization is what makes it so destructive when the factory goes rogue.
When the cellular blueprints get corrupted. And that brings us to the disease states highlighted in the cancer cell literature. Myeloproliferative neoplasms, MPNs.
Let's dissect that term. Myelo refers to the marrow. Proliferative means growing rapidly, and neoplasm means a new, abnormal growth.
The sources point out 3 distinct clinical hallmarks of this condition that you need to know. The first is megakaryocyte hyperplasia,
Which is just an absolute overproliferation of those giant balloon cells. Way too many of them.
right. The second hallmark is bone marrow fibrosis. The delicate framework of the marrow is replaced by dense fibrotic scar tissue. That's the concrete being poured.
And the third is leukocytosis, which is a massive, abnormal spike in white blood cells in your peripheral blood.
Those 3 hallmarks define the clinical nightmare of MPNs. But wait, hold on, I need to pause on that.
What's the issue?
Physically, that presents a massive contradiction. If the bone marrow factory is turning into a dense web of tough scar tissue, the fibrosis, how on earth is it simultaneously producing too many cells?
You mean the hyperplasia and leukocytosis?
Yeah, shouldn't the progressive scarring just choke off the production lines? Where is the overproduction even coming from if the marrow is being boarded up?
That paradox is exactly what makes the microenvironment of MPNs so devastating. The fibrosis isn't some separate spontaneous event. It is directly driven by the megakaryocytes themselves.
Ah, so it's an inside job.
Exactly. Because these hyperplastic megakaryocytes are mutated, they're highly dysfunctional, they undergo abnormal maturation and begin leaking massive quantities of pro-inflammatory cytokines into the marrow space.
We're talking about molecules like TGF-beta and PDGF, right?
Yes, transforming growth factor beta and platelet-derived growth factor. These act as paracrine signals. They literally hijack the normal fibroblasts in the bone marrow, the cells responsible for structural maintenance.
And they force those maintenance workers into overdrive, right? Continuously laying down dense collagen fibers.
That is the exact mechanism. The hyperplasia directly drives the fibrosis. It is a highly toxic, self-perpetuating loop.
Okay, this is a lot of dense biology, so let's pause for a quick 5-minute recap to lock this in for you. Bullet number 1.
Normal megakaryocytes are giant, water balloon-like cells that hoard DNA to rapidly mass-produce platelets.
Right. Bullet number 2. In myeloproliferative neoplasms, or MPNs, these giant cells mutate and multiply completely out of control. That's the hyperplasia.
And bullet number 3, this exact overproduction of defective cells leaks toxic chemicals that scar the bone marrow, which is the fibrosis.
Perfect. So to stop this rogue factory, researchers realized they couldn't just target the scar tissue or the downstream white blood cells. They had to find the manager giving the bad orders.
Which brings us to the core discovery in the cancer cell paper, the menin-dependent networks. They mapped out the genetic circuitry of this disease.
And sitting at the very center of that circuit board is a protein called menin.
Right. Menin is a scaffold protein. It doesn't act alone. It functions as this critical node binding to specific regions of the genome to regulate gene expression.
And in normal blood formation, the sources detail how menin tightly controls 2 very specific downstream targets, MEIS1 and MEF2C.
Let's look at those 2 targets. The foundational studies and the references establish menin-1 as an absolutely essential gene for definitive hematopoiesis.
It governs the expansion of stem cells, and it's critical for red blood cell production.
Yeah, and its counterpart, MEF2C, specifically drives megakaryopoiesis. It regulates the actual production of those giant platelet-making cells.
Okay, I want to give you a mnemonic device to help trigger your memory on this, because all these M words get confusing. Think of menin as the main manager.
I like that. Main manager, menin.
Right. And the manager has 2 floor supervisors under him. First is MEIS1, who makes the stem cells and red blood cells expand. Makes is MES1.
And the second supervisor is MEF2C, who manages the factory of megakaryocytes.
Exactly. Main manager makes stem cells, manages the factory. So menin routes the signals to MEIS1 and MEF2C.
But the researchers utilized advanced single-cell RNA sequencing to look at individual marrow cells in myelofibrosis patients.
Single-cell analysis is so cool. It doesn't just give you the average of a tissue sample. It lets you see exactly which genes are turned on in every individual cell.
Right. And it revealed that in the disease state, the blood formation process becomes heavily megakaryocyte biased.
Meaning the main manager, MEIS1, is obsessively prioritizing those giant factory cells over everything else.
Exactly. The circuitry is hijacked. This epigenetic bias is the spark that ignites the entire toxic cascade of overproduction and scarring.
So if menin is the rogue manager driving the fatal bias, the obvious intervention is to fire the manager.
Exactly. Block that node. And the source data points to a relatively new class of targeted therapies designed to do exactly that, menin inhibitors.
The literature specifically highlights two of these drugs, revumenib and ziftomenib.
These are small molecules designed to physically disrupt menin, effectively short-circuiting that central routing hub.
And the visual data from the publication showing the dual effect mechanism of these drugs is just staggering.
It really is. Let's contrast what happens when you introduce a menin inhibitor into a normal patient versus a sick MPN patient.
In a normal state, hitting that hub dampens the standard production line. You see decreased megakaryocytes, lower ploidy.
And clinically, that translates to a manageable drop in circulating platelets for roughly 20% of normal patients.
Which makes sense. It's like hitting the emergency stop button on the assembly line. It slows down normal production slightly.
But when you apply that same MENI inhibitor to the diseased hyperplastic MPN marrow,
The results are totally different, right?
completely different. It breaks the obsessive loop. You see a profound decrease in megakaryocyte hyperplasia. The runaway train of overproduction just halts.
It dramatically reduces the leukocytosis, bringing the white blood cell counts down. And crucially, miraculously almost, it drives a decrease in bone marrow fibrosis.
Yes, reversing the fibrosis.
That is incredible. I mean, scarring is almost universally viewed as permanent, irreversible structural damage.
Right, but by short-circuiting the menin manager, you stop the mutated cells from dumping toxic cytokines. The fibroblasts finally stand down.
And the diseased marrow is actually able to clear out its own dense concrete webs. It restores its own architecture.
It does. And the clinical utility of targeting this specific node extends beyond just MPNs. The researchers highlight that menin is also a therapeutic vulnerability for several highly aggressive leukemias.
The sources specifically flag leukemias driven by KMT2A rearrangements and those harboring NPM1 mutations.
Yes, the Augment 101 clinical trials evaluated these inhibitors, specifically in patients with these aggressive genetic profiles.
And importantly, these inhibitors are being tested in relapsed or refractory cases?
Right. Patients where standard chemotherapy has already failed, these inhibitors are giving them a targeted lifeline.
Okay, we are about 5 minutes from our last break, so let's do our second bullet recap to cement this in your mind. Bullet number 1.
The menin protein acts as the main manager, controlling the MEIS1 and MEF2C genetic network.
Bullet number 2. In MPNs and certain aggressive leukemias, this management network is completely hijacked, causing that toxic megakaryocyte bias.
And bullet number 3. Novel menin inhibitors like revumenib and ziftomenib shut this manager down, which reverses the overproduction and, amazingly, the physical scarring.
So understanding the biology is incredible, but I want to pivot for a second. How does this science translate into the real-world medical ecosystem?
That's a crucial point. Identifying the biology is only step 1.
Right, because there's a fascinating detail tucked away in the disclosures of the source text. It notes that one of the lead researchers, SFC, is an inventor on a patent specifically related to MEN1 mutations and their uses.
Which often raises eyebrows when people read medical journals.
Exactly. So I want to ask you, why do patents exist in this kind of genomic research? Does it restrict patient access, or is there another side to it?
It's a really vital aspect of translational medicine to understand. Identifying the NNN dependency using single-cell RNA sequencing is a huge scientific achievement.
But you can't just hand a patient a research paper.
Exactly. Transforming that microscopic biological insight into a stable, bioavailable pill-like revumenib, and then running multi-center clinical trials like Augment 101.
That requires massive capital investment, right? Hundreds of millions of dollars.
Yes. The R&D pipeline for a novel epigenetic inhibitor is exceptionally prone to failure. Most compounds never make it out of phase 1.
So the patent framework provides a window of intellectual property exclusivity.
Right. It is the financial engine that incentivizes biotech firms to take on the massive economic risk of those clinical trials.
Without that patent protecting the eventual drug, securing the funding to translate a discovery from a cancer cell paper into an FDA-approved pill for a relatively small population, it would be nearly impossible.
The underlying biology provides the target, but the IP framework provides the fuel to cross the finish line. It's an impartial reality of bringing precision medicine to the bedside.
It really is an incredible, complex ecosystem. So to synthesize this entire journey for you today,
We covered a lot of ground.
we did. We started with the sheer scale of normal blood production, examining how those giant water balloon cells bypass division to build massive genetic warehouses.
We tracked how mutations corrupt that process, driving hyperplasia and flooding the marrow with chemicals that pave over the tissue with fibrotic scar tissue.
And finally, we saw how mapping the disease cell by cell isolated the menin manager, allowing targeted drugs to short-circuit the toxic cascade and clear the concrete.
It demonstrates the immense power of understanding disease at the deepest genetic level. But before we sign off, I want to leave you with a final mind-expanding question.
Oh, I love these. Hit us.
The ability of these inhibitors to reverse bone marrow fibrosis raises a profound question. We have long viewed tissue scarring fibrosis as irreversible end-stage damage.
Right. Once it's scarred, it's done.
But we just learned that by turning off one specific genetic node, the marrow can actually clear its own scar tissue.
Oh, wow. I see where you're going with this.
Think about other devastating fibrotic diseases: liver cirrhosis, lung fibrosis, chronic kidney disease.
The pathology in those organs relies on a very similar runaway fibroblast mechanism, doesn't it?
It does. So if the bone marrow has a hidden master switch driving its fibrosis, could there be analogous undiscovered manager proteins driving the irreversible scarring in the liver or the lungs?
Are we on the precipice of an era where we don't just manage fibrotic diseases, but actively reverse them by unplugging their specific molecular engines?
Exactly. What else is waiting to be discovered by single-cell analysis?
That fundamentally changes how we view structural damage in the human body. Maybe the tissue isn't permanently ruined. Maybe it's just locked in a localized feedback loop waiting for us to find the right switch.
It's a very hopeful thought.
It really is. I highly encourage you to carry that thought with you this week. Think about the hidden networks driving the systems around us and how finding the right node can change everything. Thank you so much for joining us on this deep dive. We will be back soon to unpack another layer of the medical frontier. Until then, keep questioning the mechanics.